process of translation is part 2 of protein synthesis. So in transcription, which leads us up to this point in time, we have an mRNA strand, so messenger RNA, being synthesized from the template strand of the DNA in order for the information from the DNA to be brought out into the cytoplasm where translation can occur. So we've got mRNA out the cytoplasm, and this is kind of where we pick up from here. What I've got over on the right hand side here is an example of a codon table. Now disregard the colorings here, they're not important. All I want you to realize is that on the codon table we've got these little sequences or combinations of 3 bases. So remembering that the U's are RNA bases, so the U's replace the T's, uracil replaces thymine whenever we have RNA. So this is a codon table, and this is looking at combinations of 3 bases on an RNA strand, so a messenger RNA strand. I'll explain what this means a little bit more in a moment. I'll talk about the kind of chain or pathway that we see information for DNA being transferred through transcription and translation, and where this codon table kind of fits into all of that. So if we look at a DNA, or more closely, something called DNA triplets, what DNA triplets are sequences of 3 bases on the DNA. So if a gene is being read in transcription, then it is being read from the DNA template strand in sets of 3. So we call them a DNA triplet. So we'll have, so for example, AAT. Now, on the mRNA, so our mRNA being messenger RNA, which is created off the template or synthesized off the template strand of DNA, that AAT would then be made on the RNA strand in a complementary manner as UUA. So remembering that A pairs with T, except for when we've got RNA, we're looking at a U. So when the mRNA is being made off the DNA strand, the DNA template strand, a U is being put in place on the mRNA as it is complementary to the A on the DNA. So this DNA triplet of AAT would result in a codon on the mRNA. So we call this an mRNA for the codon. So the group of 3 bases from the DNA would be synthesized in a complementary fashion to be UUA. On the mRNA, we call that an mRNA codon. Now, our next step, and we'll talk about this in a moment, involves something called tRNA. And now tRNA is going to be involved once we read what is on the mRNA codon out in the cytoplasm. And tRNA also is going to bind to, work in a complementary fashion when binding to the mRNA. So the bases that are complementary to UUA are going to be AAU. So we're talking about AAU because we're dealing with tRNA. All right, so a type of RNA. So we don't have a T, we have a U. What you can notice about this is when we go from the DNA triplet of AAT, the tRNA anticodon, this is called, I'll write that in a moment, the tRNA anticodon basically mirrors the DNA triplet, but we see the U replacing the T. All right, so like I said, I'll pop down here. When we're talking about tRNA, we are talking about something called an anticodon. So where does this codon table come into this? So what we see is DNA, we have genes which are sequences of DNA, and they are read in DNA triplets, so sets of 3. And when they're read in sets of 3, in order to start off protein synthesis and transcription, we're making an mRNA strand 3 bases at a time. So we're building mRNA codons, which are also groups of 3 that are going to be complementary to DNA triplets with uracil replacing thymine. The next part of this process is tRNA molecules with something called tRNA anticodons come along and bind in a complementary manner to the mRNA. So we see A's binding with U's, U's binding with A's, and C's and G's working in the same fashion. This codon table over here is So it's related to mRNA because codons are found on mRNA. What it's saying is that whenever we see this particular sequence here, so we're looking at UUU, whenever we see a UUU on an mRNA codon, this is going to lead to us having a particular amino acid. Just down there, amino acid. And a particular amino acid, that particular amino acid is phenylalanine. So an mRNA codon is saying we need to arrange getting an amino acid called phenylalanine down to this site here. And the reason is because amino acids are the building blocks to make proteins, which is what we are trying to build, what we're trying to create from a particular gene. Okay? Because these proteins are going to help with structural and functional things inside of cells and with our organisms, and that's what helps make us be able to do things, act the way we do, and look the way we do. When I explain this process down below, it might make a little bit more sense. So we've got here messenger RNA. So this process here, this diagram is pretty much showing us translation. So this is part 2 of protein synthesis. This is translation here. So we've got a messenger RNA strand, and we've got something called a ribosome. Now, a ribosome contains something called rRNA, which stands for ribosomal RNA. So ribosome is this tiny little kind of organelle inside of the cell. It's this structure that is made up of largely ribosomal RNA, and its job is to go along and read the messenger RNA that was made or transcribed off the DNA is now out in the cytoplasm. So the ribosome is going to move along the messenger RNA, which contains the genetic information which was complementarily synthesized off of the DNA. So the messenger RNA is broken up into 3 base groupings called codons, which were made from 3 base groupings called DNA triplets on the DNA strand. The ribosome moves along the messenger RNA, and every 3 bases it reads, it is going to pick up this sequence from above. So it's going to pick up a sequence. Let's look at, say, so UCA, for example. It's going to move along the mRNA. It's going to see this codon UCA, and then it's going to, the ribosome is going to direct the bringing down of serine, which is an amino acid, to add to something called a polypeptide chain. So the ribosome moves along, the codon is read, and it says, righto, we need a particular amino acid brought down. What brings that down is a tRNA molecule. So tRNA, like I said up above, contains something on it called an anticodon. So if we look here at one that's already matched up, this tRNA molecule has an anticodon of UUU. This would be the codon here on the mRNA AAA. They bind in a complementary manner. So this is how a particular tRNA molecule is able to bind to a codon via its complementary anticodon. The reason why this is important is this tRNA molecule comes down and it binds in a complementary manner because the messenger mRNA carrying the info from the DNA is saying that we want in this position this particular amino acid to be brought down. So amino acids are these particular structures, particular molecules inside of cells, inside of organisms. There's 20 different amino acids that we see. So through this process, we are able to get a particular amino acid brought down, and what happens is unique tRNA molecules come down when required with an anticodon that matches a codon on the messenger RNA, and with that they bring a unique amino acid. And over time, what we see is the building of this chain of amino acids as tRNA molecules drop down and bind and bring with them their amino acid. And we get this starting to grow. And this polypeptide chain is what eventually, through some particular folding and shaping, will become a protein. Now these tRNA molecules, once they come down, they bind via their anticodon with the codon on the messenger RNA, They drop off the amino acid, which is added to a polypeptide chain. They are then sent packing on their way. We get an empty tRNA molecule, and it will go off and find and attach another amino acid, which is unique to its anticodon. Will find another amino acid and then can come down and be reused. So eventually what will happen is the messenger RNA, the ribosome will read a particular codon on the messenger RNA, which will signal to stop. The ribosome will then detach from the mRNA, and the polypeptide chain will be formed and can then down the track be reshaped into the final product, which is a protein.